Okay, Bradley, we were waiting for you.
Oh yeah. Hello.
For once, instead of waiting for me.
Yeah. Well, hello. How is everyone today?
Bradley, turn your microphone.
Oh, well, how's everyone today?
Okay.
Good. All right. Yeah. So I hear that Sushmanth has made some real progress on the Huggingface models and putting those up in that using that tool, and then he's also doing some things with the GitHub repository.
Okay. And I just learned he's also has a project on breast cancer.
Oh, okay. Wow. Yeah. Independent of the one you're doing.
Yeah.
Okay.
I have data from Kaggle actually, so I was working on it. Actually, there is a competition going on Kaggle regarding breast cancer detection.
Okay.
They have a huge data set of, and I was trying to just make a model which can predict breast cancer is there or not. So just a side project actually.
Oh, okay. We're trying to come up with a new method of detecting.
Okay.
Of making the images to begin with.
Okay.
Yeah.
Actually, Gautham Krishnan is here. I think he has kept a PR in DevoGraph.
Oh yeah, I saw that. He had a.
Yeah.
Yeah. Do you want me to go over that first? Let me go over that first, and then I don't know, Sushmanth, do you want to talk about your work that you did this week?
Okay. I will. Okay. Should I start or first you want to go through Gautham?
I'll do this first, and then we can get into that. Okay.
I think Gautham also here. Gautham, can
Hello.
you just?
Yeah, sure.
Okay. Oh, I think he wanted you to introduce yourself, Gautham.
Yeah, sure. So I'm actually a sophomore studying for BTech CS, and my interests are machine learning, deep learning, and stuff. So I've worked with various computer vision models, segmentation models, and related things. So from the past 1 week, I've been looking into DevoGraph and how it works, and I was able to run it locally. And yeah, yesterday I made a PR also.
Okay.
A small change.
Yeah.
Yeah, and I'm looking forward to working more on it.
Well, yeah, it sounds great. Thanks for participating and contributing. Yeah, look forward to it as well. All right. Yeah. So, okay. Let me go to the. Actually, I'll go to the Huggingface. I'll just preview that, and then I'll go to the GitHub. So this is the Huggingface site. It's huggingface.co, DevoWorm group. We have an organization. So, Huggingface is basically a hub for machine learning models. It's supposed to stress reproducibility, and people can put different organizations have their code up here. They have their different platforms. So, there are a lot of implementations of Stable Diffusion on here, for example, where you can download and run them. And so this is for us. We have, for now, we can add to this if people want to add. We have the membrane segmenter, lineage population model, a nucleus segmenter. And thanks to Sushmanth for putting those together, getting those up on Hugging Face. I need to create a card for this, but we might add more as we go along. I don't know. I think there are a couple more models we could add in, and then whatever people want to do. We've had a lot of activity in terms of machine learning and deep learning in the group, so I don't even know what we have available. There may be other things that we could put up here, but I'd like to have it up here because I think it's a nice place to have for people to gain access to it easily. And then we can also, I think, put other. I don't know if we can host other types of models here as well. Not really sure what the rules are. They're data sets you can host. We have some data sets we might play here.
With your permission, I just want to host these data sets and models today itself, because actually last week only I was about to deploy the data set also. I thought it would not be okay because downloading Epic dataset and keeping in Huggingface as something, yeah, I thought it may create some problem. Even Minec told me that to just take a permission from Bradly and then you proceed of keeping the whole data there. Can I create this whole data?
You mean put a data set up? Is there like a storage limit for it for the account? I don't know.
There is no storage limit for it. But I mean, we can just register as, I mean, whatever things we are doing in GitHub, we can keep in Huggingface also. There is no limit for that.
Yeah, I think we can.
Okay, by today itself, I will upload the data and I will create the model code also if it is possible. I mean, can I create? I just want to take permission from you. That's the reason I have a stop.
Yeah.
Last 3 days.
Yeah,
I just want to. I don't know, like. Can I host the whole Epic dataset here or?
Well, I don't know about host. Well, I guess we could host the version that we use. I mean, I think it's publicly available. It's not like they don't, they don't want it public. So I guess we could host a version of it here. We could also, host some of the things we have on GitHub. I can put together some things maybe we could put up here as well. So.
Okay. Is there any particular data, I mean, which DevoWorm has got, I mean, own data of DevoWorm?
Well, we put together different data sets for like summaries of the C. elegans embryo in terms of like the cell tracking positions. So like 3-dimensional position, cell approximations of cell size. And then we've done some other work with like differentiation codes and things. I'll point to the ones that maybe we should put up. Yeah.
Okay, then I will create this data set and models also I will upload the all cell membrane segmented code and all.
Okay.
Lineage population and I don't know about DevoGraph, but I can host completely of DevoLearn into Huggingface, and I will create a model card also for this.
Okay. So do you know how it hosts the data? Like when it's just something people download, or is it like render like a table, or how does it deal with that?
I download complete data from Epic dataset. Those are all CSV files. We can directly host them here. When they download it, they need to change it from CSV file to images.
Yeah.
So we can do that. And another thing was like in our GitHub, DevoLearn web is there, right? It is almost no use right now. I think we can archive that repository if it's possible.
Okay. Well, yeah, we can do that probably later. Yeah.
I mean, one issue was when I was creating workflow. I can't create workflow. Actually, DevoLearn web is using all what we call LFS space. Actually, it is taking complete LFS space to create workflow between Huggingface and GitHub. I don't have like enough storage to send those large file images or with data.
Okay.
That's the issue.
Well, yeah, we can work. Oh, go ahead.
I think we can archive that Minec DevoLearn web. Actually, that code and this code are almost similar. We can just post. We can archive. If we need in future also, we can just unarchive that.
Yeah.
I think it will not create any confusion. Like so many people are visiting DevoLearn these days. So DevoLearn web, all these repositories may confuse them because linear Huggingface models are there, which are hosted in Huggingface.
Okay.
If we archive this DevoLearn web, it will be useful.
Okay. Well, if you have an idea for how we might do that, because I don't know exactly. Well, we'll go to the GitHub repository next, and we'll talk about it. So, but we should we should talk more about that.
Bradley, I have a general question. I put a title of a book in that I'm reading.
Okay.
This is a posthumous book by Thomas Kuhn, who wrote The Structure of Scientific Revolutions.
Yeah.
You may know
it.
Yeah. Okay. The thing that struck me is that this book was written before molecular or machine learning became popular.
Yeah.
And yet it seems very much to be a contribution to what could be machine learning.
Yeah.
Okay. You might want to read it yourself. In any case, what he makes he distinguishes features. He says there's two ways of classifying things. One is to use features, and another is to use what he calls, I put it in the word in there, differentia.
Okay.
In other words, you don't look at features, you look at differences between objects.
Yeah.
Okay? And it seems to me that that's basically a difference between features, but
yeah.
though he doesn't say that.
Right.
And so I'm wondering whether he's got a new theory or he just anticipated that features would also become, that a difference between features will become features.
Yeah. Yeah, I don't know. Yeah.
I think you'd have to read the book. It's a long slog.
Yeah, I know. A
Okay.
lot of the philosophy of sciences. So.
Yes, but this is his last words.
Oh, okay. Well, we'll take a look at that. Yeah.
Okay.
Thanks. Yeah.
So I don't know. I mean, are people using differences between features as a feature?
Well, I think people are using like secondary metrics. I mean, the features are usually generating a feature space, so it's kind of like a first step. And then people do a lot of other kinds of processing.
But he's suggesting that the feature space is not important. What's important is the differences.
Yeah.
Okay. So and he was calling for a lot of research on whether or not people and animals do this.
Right.
Okay, because most of his research examples are anecdotal.
Yeah.
He really couldn't find primary research on much of this stuff, which isn't surprising because it's before machine learning.
Right.
So I think he's writing at best 1990s.
Oh, okay. Wow. Yeah.
Yeah, and somebody got a hold of his speeches and his manuscripts and put a book together.
Oh, okay.
Okay.
Yeah. Yeah, I'll have to check that out.
Okay. So you don't have a clear answer.
Not really. No, I'll have to look at it.
Okay. Thanks.
Yeah. So yeah, we were looking at the Huggingface space, and it was that we'll keep working on that. Why don't we, Sushmanth, why don't we talk about this more, like in Slack, about the data sets and everything, and what you think should be archived? Because I don't know. So this is the DevoLearn organization. So what is it that you wanted to? This one?
DevoLearn Web is there, right? DevoLearn Web.
Yes.
DevoLearn Web.
Oh, DevoLearn Web is the one.
Yeah.
Okay, yeah. I think this was supposed to be the one where it deploys on a web app.
Yeah.
So this is for. So this is out of date. Yeah, this should be archived.
Yeah. You go to settings, and at last there will be an option called archive. So.
All right. Well, I'll do that later. Yeah.
And the models which are there, which I hosted, they are private in GitHub. So just make them public also.
I can make those public. So these three private ones.
Yeah.
Okay. All right. Yeah, that's great. Looks great. Then we'll have this kind of. So we have the for people who are just coming to DevoLearn. You have the DevoLearn repository, which has this base DevoLearn software, and this was our idea. Like, this originated from our idea of building a pre-trained model for microscopy data for embryos, and we built this in 2020. We did some work a couple of years prior on kind of working on machine learning problems, and then this is sort of the culmination of that. And then we've been working on updating DevoLearn, and then also incorporating more recently this DevoGraph repository. So the idea of DevoGraph is it's graph neural networks on top of the pre-trained models, and they were nominally developed separately. There was a workflow developed last year for DevoGraph, and we're going to be working on updating that this year. And then, we want to eventually make them sort of fit together so you can use them, run them together. You can run DevoLearn or DevoGraph in the main DevoLearn package. It would just be kind of like one thing. So it looks like we have a pull request here, and this is from Gautham. So this is his pull request. So it's a fixed number 2, add animation for prediction. So yeah, we'll have. Oh, say I guess I'll review it later. I haven't reviewed it yet. I didn't really get to it, but yeah, I'll do that.
Those were presumably the centroids that were marked in the cells.
Yeah, this is this is from like one of the things that gets segmented in DevoLearn, the base of DevoGraph is DevoLearn, just the segmentation part, and this is of course the segmentation. So this is segmenting the membrane edges and the centroids, which are roughly the membrane or roughly the nucleus, but we can't be entirely sure.
Stupid question. Has anyone investigated the distance between the centroid and the center of the nucleus?
I'm not that I'm aware of. You mean the centroid that gets generated? Yeah.
Yeah, centroid is generated in the actual center of the nucleus.
Yeah, we haven't done that. I mean, I don't know if people have done that as a study. Yeah.
Actually, they haven't done that in the DevoGraph. They just generated centroids and they tried to create a graph neural network out of it.
Yeah.
Okay, the reason I ask is that I've been reading about diatoms, these nuclei a little bit, and they move around a lot.
Right.
Okay, so they're probably frequently not in the centroid itself.
Right. That is a problem though, yeah, with movement, because we're just showing the static versions here. We usually calculate centroids for each frame, so it like changes per frame.
It might be an interesting study if you have any data set where you can get both the membranes and the nuclei.
Yeah.
To compare to see how well the centroid predicts the location of the nucleus.
Yeah. Yeah, I know we had a problem with some of the data that they have, and I don't know what the latest data is, but like in staining the nucleus, getting a good stain of like the nucleus that's sort of localized. It's just kind of like they put a stain in the nucleus and it's kind of all around it, and it's really hard to get a handle on where, like, the nucleus could be, if you're talking about segmenting a cell, it could be quite a bit of the cell, up to like 5 or 10% of it in the middle,
Yeah, so it's hard to get a data set where you can see both the nuclei and the cell membranes.
Well, I mean, I guess the thing you'd need would be a very accurate nucleus stain, or to like find have a nucleus stain where you could find a centroid for the nucleus, and then a centroid for the whole cell body, which is actually something that could be calculated from the membrane, I think, because you could find So I mean, you could just do it that way, and then because I don't remember. I think the centroids are usually kind of a safe way of saying nucleus. We don't want to like assume that it's the nucleus, but yeah.
Okay, I'm just saying a quantitative study questioning that accuracy might be useful.
Yeah.
Okay.
All right.
And if it could be done in vivo, then you could track whether the nuclei are moving.
Yeah. Yeah, that would be great.
Actually, the model is not working well when there are more cells. DevoLearn is also not segmenting properly. When you check for this Gautham Krishnan PR only, when you run that slider, when cells are densely populated, it is not creating, like, this is an example, but Gautham Krishnan have another video of it. When the cells are densely populated, we can't get clear segmentation of, centroids of all video.
Yeah.
Gautham? Hello?
Yeah, can I just share that?
Share
Yeah,
that.
let me stop sharing my screen. You can share your screen.
Okay, so this is the actual cell membrane segmenter that is working, and it's working as expected. And instead of using the cell membrane segmenter, we are also trying to use the nucleus segmenter on the EPIC dataset. Okay. So we can see when the number of cells are less, it is predicting accurately, but after some time, the predictions are way off.
See, these centroids are not working fine, and the segmentation was not clear at all when cells are densely populated.
Yeah.
I think we need to work this on this year. Segment it clearly. We can use DevoLearn, but on top of it, we can use other methods also to segment it for when the cells are densely populated.
Yeah.
Can I make a suggestion that these pictures are excellent, but can you make them more informative by using a different color for the centroid of the membrane and another color for the centroid of the nucleus?
Yeah, we can do that. We can do that, but by next meeting we'll show you, like, for cell nucleus there will be a different color and the membranes will be different color. We'll try on that.
Yeah.
Okay, we make it much clearer.
Yeah.
It's not obvious what one is looking at.
Yeah.
So also, I just want to show another thing. So in the cell membrane segment in the EPIC dataset, we can't actually get a good prediction. Right. So the thing is, in the cell membrane segmenter, the video, this is the input video that we are feeding into the model. Mm-hmm. But for the EPIC dataset, this is the video. So
actually, Gautham,
yeah.
What happened was, like, if you give this video, it is not working fine. When you change this video into a grayscale image, and then you just need to give it to cell membrane segmenter, then only it works fine. Actually, it is the main issue thing. You need to change this into grayscale, and then you should need to give it to cell membrane segmenter. Then it will work fine.
Okay. Oh yeah, that makes sense.
Is the green is mostly intensities of the same wavelength?
Yeah, it's a fluorescent stain. I think it's just like in the EPIC data set, it's like, I think it's like a nucleus. Well, it's not clearly localized to the nucleus, but it's basically a stain in the, somewhat localized to the nucleus for like GFP stain for some factor, and that's expressed across cells. So it's like not differentiating in terms of depth, or
it's a fluorescence image, so it's in a single wavelength.
Yeah.
Okay, so the intensity is just brightness in that wavelength.
Right. So the less intense ones, yeah.
I just want to make sure I'm not missing trouble.
Right. Actually, I think the less dense ones are farther out. Like if you think about a 3D volume, the less dense ones are in a different focal plane. But I'm not sure exactly how they captured it. So.
Well, it depends on, is that a sectioned image or
yeah, I think so.
So it might be that they just didn't they only caught the top of the cell.
Yeah.
Actually,
top of the nucleus.
Actually, when I was reading about this data, they have captured using ultraviolet rays. They emitted ultraviolet rays using fluorescence, something chemical called fluorescence. They added to that whatever elegans is there, and they emitted ultraviolet rays on it, and then they captured this video, whatever it is there.
Yeah.
Actually, in this process, it has been captured.
Yeah. Yeah, I don't know if they're like the reason we use the and the EPIC data set is quite old actually now. It's probably about 15 years old, so it's been around a while, but it has like the most complete accounting of the developmental process. There are newer techniques like SPIM imaging and we use some of those images for the other where you see the membranes, training for membranes. And those are better. Those are a lot higher resolution. the stains are more distinct. They've stained the membrane and the nucleus or something approximating the nucleus. And then, you have those data, but we're kind of putting those together into one model to get what we're doing with our predictions for DevoLearn. And so it's going to be kind of hard to get things later on in development. I mean, there are probably ways to do it, but I couldn't tell you exactly what to do. It's like one of these things where it's trial and error really to get the best outcome.
Okay. Actually, I have one paper I'm sharing in the chat box.
Okay.
I was thinking to implement this for segmentation only. This is working fine actually. I mean, I have shared. I hope you got in the chat.
Yeah.
Can you see?
Yeah. Oh yeah.
Yeah.
Okay. Yeah. This is establishment of morphological atlas of C. elegans using deep learning based 4D segmentation. So let's see. Yeah, this is the one here. Yeah, I think I saw this at one time. So this is where they've kind of, let's say, they're kind of characterizing a lot of these processes. So in the different data sets, they don't really characterize the processes. I mean, they visualize them, but they don't really characterize them. This is putting together like an atlas of C. elegans from, for this whole process. And then they create, so here we're reporting automatic pipeline CS paper, which combines automated segmentation of fluorescently labeled membranes with automated cell lineage tracing. So what they'll do, actually, that's what they've been doing that's what you have with the EPIC data set. There's actually separate metadata that tells you the different cells, like there's an identity of the cells that's characterized by the lineage tree. So every cell that gets generated in the developmental process, when there's a cell division, it has a certain identity. And so this is what they're doing here. They're putting this together. They're generating a time-lapse 3D atlas of cell morphology. And so they have this, I don't know kind of what the outcome of this was. I can't remember. This is the pipeline though. Oh, go ahead.
This is the outcome part. They have mentioned it, like they have mentioned the clear images of it, like how it will be 4D segmentation video of it.
Okay, so they just have some benchmarks, and for segmentation results, they have volume inconsistency, which is not. I mean, we ran into this problem where there's inconsistency in the volume of the cells that are. So when you generate a cell through segmentation, you get inconsistent volumes, you get lost cells. It's just what happens. Then they have this morphological atlas. I'm just wondering if this is something that is online. I didn't see the link. But yeah, I remember this. This is like a 3-dimensional model of the embryo, and then they have this. So they can do this.
Do they reconstruct the tree?
Yeah, they do reconstruct the tree, sort of from these images. So they have this tree sort of in the 3-dimensional space here. So it kind of looks like a network, but it's really just connecting the different cells of different. So like, you have the AB lineage here, different colors.
I think they said they were following it over time.
Yeah. So they're doing this in space. This is in spatial coordinates, but they're doing this in time.
Okay.
And so,
do they end up with the same tree as the original one that was done by hand?
Oh well, I mean that's what they're aiming for. I guess they do because they train it on that.
Was it white?
It was Solston.
Solston.
Yeah.
Solston.
Okay. Yeah. So they actually look at cell contacts here, and they have these criterion for the different features of so there's cell contacts, there's cell shape, and they have to approximate all these things. And then this is cell shape irregularity.
Oh, there it is. Okay.
Yeah. So this is the tree. It's interesting that they have this cell shape irregularity here, as we've talked about, I think, cell shapes in, yeah. So you have these different shapes.
My question is that tree the same as the Sulston tree?
Yes, it is.
It is. Okay.
Yeah. I mean, they're basically using it to train the algorithm to build a reconstruction based on the tree. So they're using the tree, and we do this too, is to train the model based on the tree.
They're putting in the result they want to see if they get the result they want.
Well, no. No, it's just that, I mean, you can clearly see the divisions. It's like you can ID the cell based on its position. It's not like, C. elegans embryos are just unique in that way. So you can just basically say, this is what that cell's supposed to be. I suppose in some case, maybe we haven't done, maybe the tree isn't correct. I don't know. But in most cases it is, because we've been able to observe this a lot.
As I recall, in some cells in C. elegans, you get a pair of cells that are identical.
Yeah. Yeah, the sister cells. Yeah,
do the sister cells always do the same thing from one embryo to the next?
I think for the most part, unless you're dealing with mutants. In mutants, sometimes cells don't divide in the same way. Sometimes you lose cells.
Yeah, but I guess what I'm saying is if you've got a pair of cells that are the same kind,
yeah,
is the subsequent trajectory of the two cells always the same?
Yes, as far as I know.
As far as you know.
Yeah.
Okay. Okay.
I suppose it could differ. People have made observations on this, and they say, well, it's a very predictable tree. It's not entire. I mean, it may be that that's actually not true. But
okay. Sorry, I'm being a gadfly.
Well, no, I think that's a good question, because that's an assumption we all make in C. elegans, is that it's identical, almost like a machine. And I don't know if that's really true, because in mutants you see differences.
Yes, in mutants you see differences,
right.
And I'm saying, look, we have the data that identical twins are never identical in humans.
Right. Yeah.
Okay. So when you get twinning in C. elegans, do you get exactly the same thing every time, or is there a variation?
Yeah, I mean, there may be a little bit of variation, like stochastic variation, but otherwise you do. Yeah.
I know, but what?
Yeah. Yeah, I think, my answer, I guess, would be, I think it's pretty much the same, although you do see a little bit of variation, maybe. And that may not be true, all the way around.
Can you extract out of all this data the actual trajectories of a pair of identical cells from different animals? That would be the telling thing.
Yeah. Yeah. I'm just writing some notes here.
Okay, sure.
Yeah. So this is I'm going to go over this graph or this figure. I'm going to move on. But I just wanted to show this. I thought this was interesting. So these are these different idealized shapes that they have here. And then they have the tree. And then what they find is they find these different cells. This may speak actually to your question. When they segment the cells for like these different cell identities, they have different, kind of slightly different shapes and sizes. So for ABPL, which is a cell in the AB lineage, ABP sublineage, which is, I think it's up here, ABPL, you get these different shapes. So you don't always get the same shape. It's a little bit different each time. And they're composites of these different types of shapes. So these are the sort of the platonic shapes that you would expect, a spheroid, icosahedron, and so forth. And then these are like the combinations here where you get different things that come out. So you're just segmenting membrane.
Okay. Are any of these cells the kinds that produce a pair of identical cells?
I think ABPL does. ABPL splits into ABPL A and ABPL P, I think, anterior and posterior. And they're identical. They just move to different parts of one to the anterior end, one to the posterior end, and then they.
Okay.
Yeah. But again, they're going to look different, as you So MS, you have differences in that. You have differences in ABAL and then P3. And so this is, yeah.
Okay, I guess I'm asking a broader question, which is is there something that could be used here to measure what we might call global control of cell positions?
Oh, yeah. You mean like
words,
positional?
is there actually a global control of any sort? Or is it all local?
I think I think there is global control for C. elegans because you have cells that go, that's a good question actually, because you have this initial anterior posterior division, and then you have differentiation there, and then you have divisions within those hemispheres, anterior posterior, and they're going either left or right or anterior posterior, and so there's local division and there's localization there. So you don't have things that move all the way across the embryo, but there is cell migration. So there may be some local, I think the positioning is local, but there may be some global things going on in terms of organizing things. Yeah.
Yeah, this might be a way to measure that.
Yeah.
Okay, because that's one of the outstanding questions in embryology. Okay, do global controls actually do anything?
Yeah.
or is a local model good enough?
Right. Okay. Well, I'm going to move on from that paper, but thank you for bringing that up, Sushmanth.
Okay.
And then I just wanted to go over quickly, if people are interested in this project, this is the GSoC project for this year. This is on the INCF NeuroStars, which is their discourse. This is where INCF, our sponsor, has posted all the projects that they're sponsoring. This is 3.1. So if you go here, you can ask questions about the project. And then I've also provided a link to the Slack. So if you're interested in joining our Slack here, you can join via LaunchPass. The Slack is a better place probably to discuss some of this, but this is also a good place because you'll get feedback from INCF as well if you need. So we have some interests already from people. Arnab actually is working for INCF as a sort of a coordinator for their GSoC project, and he was an intern in this group back in 2018. So he was one of our students, and then he became involved in INCF's GSoC program, and so he's spreading the good word of DevoWorm to the rest of the world, I guess. Great. So I think that's great. Thank you, Gautham and Sushmanth, for that. How's Susan doing?
There we go.
Yeah.
I'm doing fine. I have a presentation on some of the stuff I presented to the group this week for my course. So I'm, I made another PowerPoint.
Okay. Would you like to share it?
On Tensegrity.
Yeah.
So would you like to see it?
Yeah, I'd like to see it. That'd be great.
Well, I can show you just parts I haven't shown you before, maybe.
Okay.
Okay. Share my screen. Okay, I'm not sure. It says select tab, select window. There. Okay. I don't know.
Here we go.
See what I'm doing.
Little library.
There. This
Okay.
is my presentation. And I'm discussing optical coherence elastography. Can you see this?
Yeah, I can see it.
Okay, and then tensegrity and the definition of it. Epithelial tissues, because that's what I'm modeling, and then my modeling of epithelial tissues, and then some examples. And I showed the examples the other week.
Yeah.
So I start off with optical coherence elastography, and this is a basic steps in the elastography. You take an image, a 3D image, using optical coherence tomography of a before loading and after loading of a tissue or whatever you're measuring. And this is an image of doing that. And then you make a map estimating the mechanical properties of what you've just taken images of.
So it's like you push down on it and then you take an image and then it shows like the,
Yeah. And you don't necessarily have to push down on it. You can pull on it as well, but most of it is compression imaging.
Right.
Anyway, this is optical coherence tomography setup in Dr. Sharif's lab. So it's a swept laser source, and this is all infrared light. And the central frequency is 1100 nanometers. And I think it's plus or minus 100 nanometers. Don't quote me on that. I need to look it up. And you can. The result is here in blue. It's that's your raw data, and then it's made into a signal here. I think using a GPU setup. Not sure about that either. So I shouldn't say anything. And this is a salamander egg underneath the microscope apparatus. It's over here in the sample arm. This is our sample arm here in the lab.
Yeah.
Okay, and this is the heart of how it works. You have a sensor layer and a tissue layer, and this sensor layer is made up of fine layers of let's see, gelatin or elastic material that kind of matches the tissue. And when you get your image, all of this blue layer in the sensor matches this blue layer in the tissue. So you can say that the elasticity of this layer matches this layer in the tissue. So this tissue elasticity is the same as this blue layer you've got here in your sensor. And the same with the red layer and a red here.
Susan, what is the gray bar on top?
Up here?
Yeah, well, what's the gray?
That is, in this case, this is the compression and imaging element. Like you're pushing down with this.
Is that visual optics?
Yes.
Yes, okay.
It's infrared light. And
infrared optics, okay.
Yeah, and it's microscope, basically.
Okay.
For infrared light.
Gotcha.
Yeah, and they're showing the color bar here. Like blue is, I'm not sure, negative. This is positive, but I should probably look that up. Somebody's going to ask me about T and TT. Yeah, okay, look that up. Minus TT. And what is TT? Anyway, it's pseudo-colored.
Yeah.
Anyways, this is an example of something that's optically similar. The inclusions are optically similar, so you have an optical coherence tomography image and you don't see anything, But if you use optical coherence elastography, then you can see the stiffer inclusions. So and you can use this for live histology imaging.
Wait, what's the spatial resolution?
Spatial resolution? It depends, let me go back here, on, oh, you mean, yeah, I don't have a scale bar in this. They didn't have a scale bar, but.
Well, it says it's about a millimeter, what's a millimeter across, but the edges of the objects look a little bit blurry, so obviously there's some limitation on spatial limitation horizontally and probably vertically.
Yes, it depends on these sensor layers and the difference in elasticity between the layers, because you're only going to try to look at the image of the sensor layer and compare it to the tissue. So it depends on how fine the difference between these sensor layers you have. Like,
okay.
This is maybe part of why they have me searching for Tensegrity to see if I can make a more refined model of this.
Okay.
Might be part of it. Maybe I'm supposed to be making a better sensor layer somehow. I'm not sure. Anyways, so then I go on into Tensegrity, and I think I've shown that here before. Tensegrity is a structure that has compression elements. Here they're in green and tension elements in red. So I've shown you some of this before. This is Tensegrity is multi-layered in living systems. So this is actin, this is cell cytoskeleton, and they appear to be a tensegrity. And cells have tensegrity components and tissues. And also when you get into larger structures such as the lumbar spine, the bones and ligaments act as a tensegrity system.
Okay, Susan, could you hold that slide a second? Bradley, I want you to look at this carefully.
Yeah.
Because this shows that the nucleus is positioned by the cytoskeleton.
Yeah.
And it may not necessarily be at the center of the cell.
Yeah.
Okay? And
Right.
if the cytoskeleton starts changing, the nucleus can move around.
Right.
Okay, anyway, this,
Okay, go ahead.
all right. I'm just, this is long, it's like 30 slides. Anyway, this is the tensegrity I'm using to model my systems. And here is an example of stress strain curve that is for a living tissue. And they have sort of a J curve where you have a toe region that's a different slope from the elasticity region. They've got an A here. And then, of course, it gets into the plastic region where it starts to permanently deform and then fail. This is the simplest type of tensegrity structure you can get. And they've done some stress strain curves with it. And this is, if you have elastic string, you get these types of curves, which are not that J curve that you need for a tensegrity structure or for a living tissue. So if you have rigid cables in compression, you get this J curve that they're that you get up here.
Susan, I've got a question about this curve here. Go back to your failure curve.
Which curve? This
The
one?
Troy failure, yeah. There's a famous spoof paper. I could look it up. many decades ago, where if you put flies in a box and heat it up, you get a sudden phase transition.
Yeah.
Okay, so I want to ask, when you have failure, are those dead cells?
I don't know. But I know you've done some experiments with axolotl eggs where you compress them
Oh
for
yeah,
a while and then they were permanently
that's right. They're permanently deformed,
deformed.
but they were still viable up to a point. So
Yes. Well, that would be my guess, is that when you get over into failure, then they die. There you go, there's death.
maybe, or if you put them back cultured, they recover.
Right.
In other words, in a way, is there hysteresis in this curve?
In some tissues, there is hysteresis, but in something like ligament, usually there isn't.
Okay.
So it just depends on the tissue. I have a breast cancer curve that has a very small hysteresis.
Okay.
So I'm
Okay.
just going to go with it. Depends on the
Well,
tissue.
you have to find out if failure is death. I'll dig up that reference and send it to you guys.
Yeah.
That's funny. Okay. Anyways, so then I go in to epithelial tissue. So I'm modeling something that can be a simple one cell thickness that's on a membrane. It's a basement membrane or extracellular matrix, depending on what you want to call it. So these can be flat or in between or tall and columnar. And then I could get into stratified layers because there are some things like, I guess, the urinary tract where they have stratified epithelial tissue.
Is columnar that is fully stratified?
Columnar?
Columnar at the bottom. Oh, I see. Stratified. Yeah,
It's
okay. I see.
stratified, and there's more cuboidal
3 layers.
cells
So
on the bottom and then columnar
this
on the
would be
top.
like skin and dermis.
Yeah, it just depends on where it is in the body. I know in the ear you get this type of thing where you have actually bone here, which is different from skin. Yeah, and I'm not. Skin tends to have. Well, it's complicated. Skin has a complicated structure. It isn't just.
Oh, so it's not simply columnar in the stratified category.
No, it's.
Okay.
No, it has some of these. Well, I don't know. I get which one you would say skin is. I think it depends on where the skin is, but it's at least 5 layers thick.
Okay.
And I had an image of where each of these was in the body, and I would guess that this would be skin here because it has this layer on top of dead cells.
Okay, which would be called dermis and epidermis?
This is the dermis, and this is the epidermis, pardon me, and this is the dermis underneath.
Oh, underneath. Okay.
I would think so, yeah.
Okay. The reason I ask is because I used to study melanoma, and one of the big factors was that if you caught it early, you could cure the patient 95%. But if if the melanoma penetrated, then death rate was 95%.
Yeah, so if it gets beyond this, the ECM, extracellular matrix, this thing here, right, the basement membrane,
Right.
then it causes problems.
Does this mean the blood vessels don't go above that layer?
It looks like there's blood vessels in the dermis and not in the epidermis. Yeah, it does.
Okay. Okay. Thank you.
I don't know. I'm not sure how these are kept alive. They're not exactly. Some of them die off at the top to help keep your skin secure.
Yeah.
And if you rip that off, it hurts. this upper layer. Anyways, I'm not a dermatologist or a medical doctor.
Yet.
Just saying. And then I showed this last week. It shows that actin and myosin tend to form a structure on the outside of a cell. And then this is a look at the top view of an epithelial tissue. And you can see that it's attached at the corners with the actin and myosin. So this is kind of the structure I was going for with my modeling. And here's a side view in a cartoon. And you have the actin-myosin complex here. Yes, that's this is actin and myosin. It says actin cytoskeleton, but there's also a myosin component to it. I'm not too sure about my cartoon here, but anyway, there's cadherins that join the cells together, and there's integrins that hold it to its basement membrane or its extracellular matrix along the bottom.
When do you see microtubules at the apical end?
I'm not sure, but what they were showing was that there was actin at the apical end here, and then they showed that there was microtubules and intermediate filaments holding the nucleus to other parts of the cell, to the desmosomes and to the integrins.
These are adult cells.
Yeah, these are cells that aren't changing that much. They're adult cells, yes. So from a previous slide, you could see that there are microtubules at the surface. So again, I assume it depends on the type of cell and its maturity.
We were like in the last section, we talked about C. elegans and the developmental cells and the atlas that they're building. And that's one thing we really don't have for like developmental cells and C. elegans is like the stains for like microtubules and some of these cell junctions. I mean, I don't know, the data for that is pretty sparse actually, but
I would really like to see that because it certainly doesn't help me make a cell model if I don't know what's going on. And they say, oh, no microtubules are involved. And I'm going, yeah, sure. And you take the microtubules out of a cell and it just dies.
Right, yeah.
Sure.
Well, I imagine in development it's much more dynamic. Like the network is shifting a lot more, especially when cells are dividing and things like that.
My question, I guess, is in these adult cells, to what extent could we regard the apical structure as being the remnant of the cell state splitter?
Yeah, don't know. Not enough information, and I've looked for this. Like I have piles of papers here trying to find it, and it's just trying to find.
The original paper is one by Beth Burnside. Maybe a follow-up to that paper might help.
Okay,
Beth Burnside. If you have trouble identifying the paper, let me know.
Okay.
course, didn't use the term cell state splitter, but she observed apical microtubules that are parallel to the apical surface.
Okay, well, there's certainly apical actin, actomyosin, and actomyosin at these junctions.
Okay, yeah, but okay. So a follow-up of her paper might uncover something.
Okay, sure. I don't mind. Here's your actomyosin. Here's your myosin, and here's your actin, and actinin. I don't know how you pronounce that. Attached to a focal adhesion, which in turn is attached to your integrants, to your extracellular matrix. So what you need for your your model, you have to have pre-stress in your cytoskeleton, and you need the extracellular matrix adhesion through these integrants. So it's not so much dependent on the elasticity of your cables. You've got to have pre-stress, and you've got to have attachments. And it was pointed out to me by someone named Stephen Levin that I was forgetting time. And I'm aware of this. You have relaxation and creep in tissues, and that's often measured rather than this stress strain diagram. And tissues stiffen faster if you have a faster force applied to them. And tissues also stiffen more if more force is applied. And I was wondering if I should put that picture of actin showing more actin accumulating if there's a larger force. That's proven. So I'll just show you what I did for a model. So here's my model. I just made it a hexagonal model, and I did it in 2 parts. So this is the actual actin ring that I'm kind of simulating here, just as a tensegrity structure. So it could be microtubules as well, because there's a stiff element as well as the strings. And then I've got posts around the outside. And when I attached things together, like the posts to the cell model, I tried a plane attachment, and I tried a tensegrity type attachment. And guess what? The tensegrity type attachment works better. For the plane attachment, I got minus, what is that, 6 times 10 to the minus 5. And if it's in tension, it's 6 times 10 to the minus 4. But if you have a tensegrity attachment, you have 6 times 10 to the minus 3 newtons that it will take before it falls apart, and then 6 newtons in tension. That's quite a difference. It's factors of 10. So no, 100, I guess, in compression. And I did finally get this to work with 3 cells. And this structure tends to put the largest stresses on the outside of itself, on the posts. It just does. So there you go. And you saw these examples last time. So
Yeah.
how long did that take?
Oh, yeah, it's fine. Probably about 25 minutes, 30 minutes. It's fine. It's great. That's great. Actually, I had a question about the forces. If you, like, I think it's 27. So slide 27.
27.
Just the next one
okay.
up, yeah.
Oh, here, yeah.
Yeah, so you say compression point forces in z direction max, and then negative 0.0006 newtons. So negative newtons would be like a compression force. A tension
Yes.
force would be positive, or
Yes.
okay.
Yeah. So it's less flexible in compression than it is in tension, but that's maybe partly because I had the model, well, really attached to its base, and its base is not flexible.
Oh, okay.
I basically had it pinned.
Yeah.
So
it goes down, and you can't push it down very much, but you can pull it up a lot just from the
yeah,
base.
so I had these posts pinned.
Okay. Yeah.
And in this model, I actually just had the outer part pinned.
Okay.
And so all of the forces ended up on the pinned section,
Yeah.
right here.
Okay, stupid question. If you extended this model so that the plane of the three structures there becomes much larger and you bend it into a sphere, then what happens?
I just got this to work.
Yeah.
No, that's
Yeah, I know. It would be neat to put this in a sphere,
Yeah.
a spherical configuration. I agree.
because then the posts all of a sudden are not a discrete ring of posts.
They're not discrete posts.
No, they're not a ring. See, you got a ring post on the outside, which are taking most of the force. But if you bend the whole thing into a sphere, then the ring disappears.
Yeah.
In that sense.
There's a positive curvature of the surface of the.
Yeah, then it'll have a curvature to it. Yeah.
We curved into a sphere. So what happens then?
Now that would be an interesting question.
You might need the university to buy a much bigger computer.
I'm already getting complaints. You put too many components in this thing. It must be a lot.
Oh yeah.
Yeah, this took me a whole day to put together because of the little tiny, little itsy bitsy parts.
Yes, but your time is
in here.
They don't pay for your time.
No, they don't. So I should say, what are you complaining about? You didn't have to pay me.
Right.
That attitude.
Yeah.
Hey, my attitude's always been computers are never fast enough.
Oh, well.
Yeah. Well, thank you, Susan. That was great.
Anyway, I have to give this on Thursday in about 25 minutes. So.
Okay. Yeah.
I'll be showing them this part.
Yeah. Oh, yeah. Okay. That part too, or?
I showed you this last week.
Yeah.
Where you have substrate elasticity changes stem cells or mesenchymal stem cells into either neural tissue, which is blue, myo, which is muscle in this green, and an osteo, which is bone in this orange color here, depending on the substrate elasticity.
Right.
So your basement membrane or your extracellular matrix, depending on what you want to call it.
Yeah.
That's very unscientific. Do I need to be more scientific when I present this?
Yeah, that's fine. Yeah. I think just showing the difference. I think that's always what they want to see. If they see a difference, or if you can explain, like, if something looks different, that's usually what people ask about. So.
Okay. Well, I've got that, and then they may make me take this embryology out. I think it's very interesting.
Yeah, it's in context, right? It's like, well, why do I care about this? Oh, yeah. Well, actually, in engineering, you might care about it. But...
It wrecks an embryo.
Yeah.
Wow. Anyway, there. So that's my presentation. Anyways, it's too long and has to be done in 25 minutes.
Yeah, I mean, you just learn, like, to race over certain things, and, and like, it's not the best in the world, but like, you have to learn how to do that sometimes, just to press it down and get it.
I
I mean,
got
just
myself tilted.
yeah. So yeah, it looks great. I think you'll do well. That's a really good presentation. You've got some good.
Thank you.
Yeah.
All right. Well,
All right.
thanks for letting me try it out.
Oh,
I
no
need
problem.
to
Yeah.
rehearse it again one more time.
Yeah. All right.
I'm going to go to the APS meeting, March meeting in
Okay.
Las Vegas.
Oh,
What
wow.
is
Okay.
APS?
APS Physics. It's a large
American Physics
society.
Society?
Yeah.
Okay.
So I'm going to go to that meeting and attend the soft matter lectures.
Oh, okay. See if you can. I'd be especially interested if anyone. There are a lot of physicists modeling balls of cells, and the cells are usually of uniform type. You want to know if anyone has gotten up to 2 cell types, let alone an embryo.
Oh, I'll see.
Okay. So let me know if you see anything like that.
Okay.
Okay.
Yeah, I know people have been doing things with like embryoids and other things like that where they're really.
Oh yeah. Yeah.
Yeah.
Physicists seem to be afraid of whole embryos.
So, are you looking for modeling with more than 2 cell types, or are you just looking
At least 2 cell types.
for the modeling?
Yeah, like the onion structures. You remember the old onion structures from the differential adhesion days? No.
Yes, differential adhesion and the cells tend to go around each other if the surface.
They made so-called onion layers, and then they would work out this kind of cells that sticks to this kind of cell better than to another kind, and
Yeah.
okay, they got that far, but that was before there was any real modeling. So
Oh, okay.
it was all based on differential adhesion. But the physicists are getting much more complicated models, but they only go to 1 cell type.
Okay. All right. Look for differential adhesion and surface tension and see if somebody's got something going with more than 1 cell type.
Right.
Okay. Yeah.
Right. As I said, the physicist approach is giving it a spherical cow.
Yeah.
Oh well. Okay.
Right.
I guess it's good.
All right.
Anyway, thank you for letting me show you that.
No problem.
Okay,
Yeah. Thanks. Oh, you're still sharing your screen. Yeah.
Yeah, I need to stop sharing the screen, and it's got stop sheets.
There we go. All right.
There. Thank goodness.
Yeah.
Finally.
After you've cleaned up the slides a bit, like putting in an explanation for TT, why don't you give a copy to Bradley and put in the archives?
Yeah.
Okay, I'm not sure what Dr. Sharif would think of that, or if it would be okay if I could share it. I'll ask him if I can share it, and then I'll
Okay, fine.
yeah, I need to ask him. He has some notions about things.
All right.
Ask if share for Sharif. Yeah, his first name is Sharif and his last name is Sharif.
Okay, Bradley.
All right. Yeah. Well, yeah. Okay. I think that's it for today. Any other things you need to mention, or?
Yes, you have time to go over our paper.
Yeah. Actually, I could do that real quick. Do you have time?
Oh yes. Yeah.
Okay. Yeah. Let me share my screen. There we go. I have it up. So I've put this into this is the simulation that I've been working on, and I put this into the main paper. I'm not done yet, but I'm getting there. So this is in the main paper. This is the paper that we have been working on, and I did an earlier contribution to this. Nick's done some things with droplets, oil droplets, toy model for the origin of life. So the whole point of this exercise, these simulations, is to model the origin of life, and so by spontaneous means. And so there's some literature review here, a model of autocatalysis, which we talked about last week. We explained what that was, and then getting into some tables for that, and then coming down to I put in, or there's a part here on applying Braitenberg vehicles or Braitenberg-like vehicles to this problem. So this is a part that I contributed earlier, where you would create Braitenberg vehicles. So Braitenberg vehicles are these toy models where you would put together a very simple organism with a nervous system, but it's using this analog of vehicles with wheels and a body and light sensors and things like that. So this is sort of a diagram of the model. You have these viable and inviable Braitenberg vehicles. You can put together the pieces, and the idea is that they assemble assembling a 747 in a junkyard using a tornado. It just kind of comes through and it pushes the pieces together. And if you do this enough times, you get like things that are functional. So that was I think it was Frederick Coyle.
Now we know what you get. You don't get an airplane. You get a balloon.
Yeah. So so this is just kind of I worked this out as a set of viable and inviable vehicles, giving examples here using the toy model, and then coming up with so having like a parts list with a number of different states that the parts list can take, and then the probability that will result in a spontaneously functional vehicle. And
I was surprised you got over 40%.
Yeah, there are actually a lot of configurations which end up being viable. And this, of course, is just based on my toy model, so it could vary. But yeah, it's I think it's surprisingly high. So yeah. So anyways, using a focused methodology, you can get that result. And then the second part, which is the part that I've been working on. So this is computer simulation of a vesicle. So I need to change some of this, but this is basically the parameter list. I need to finish this up. I think this is most of them. This is just taken from I'm running this in Scilab, which is sort of a version of MATLAB that's open source. So it's that type of language. It's a symbolic language. It does a lot of matrix calculations and that sort of thing. So this is just setting up the parameters, and then the graphs. This is the idea of this molecular world with a lot of infinite number of molecules of a certain type, and then having a cell of a certain diameter where these molecules get put into that cell, or the cell membrane the vesicle membrane is more or less permeable, but you can only fit so many molecules into the cell at a time, and so the chances of getting K-types is, as you increase the diameter, it increases. So this is something, and the idea here is that if you have to get to 36 types to get spontaneous autocatalysis. So for small membranes or small vesicle diameters, you can't do that. For large ones, it's automatic. But the question is can you bootstrap this in a very small vesicle from something that has fewer types to more types? So I showed this last week in the code, and I've been working on this. I've been working on a sort of another version of this where there's like this longer duration of simulation. So this is a simulation for 10 to the 5th days. So everything in terms of this graph actually shows how this goes for like two days where there's a day-night cycle, and how basically you get a lot of variation during the day. At night, things kind of close off, and there's this resting period, and then you get this fluctuation back. And then, but that doesn't necessarily result in any sort of increase in the number of molecules. It's just very static for a vesicle of a certain size. But one thing you can do is to actually, I did two things in this. So this is the first piece of code that's not really that important right now. The second piece of code is the more important thing. This is the one that I was working on this week. This is for 10 to the 5 days, so I can't run 10 to the 6th. Well, I maybe could run 10 to the 6th, but I'm hitting the upper bounds of my computational limits here for what resources that I have, because you're generating a lot of characters or a lot of variables for this. But so you get up, like you can just take 10 to the 5 days. Each point is 1 day that has like a mixed day-night cycle, so it's like, you know, half of the time is day, half of the time is night. And it can change that in the model. What we do is we draw from a mixed uniform Poisson distribution. So that cloud I showed you in the previous figure, that cloud, it had it like it was a bunch of particles or a bunch of molecules of a uniform distribution. So that means that any one type is just as likely as any other type. And so that's what you have. But in this case, some types are a little bit more likely. So you have this Poisson process that's mixed with the uniform distribution. The Poisson process introduces this non-regularity, where it brings in more types at different frequencies, and in the base, the base sampling pool. So that means that the higher this arrival rate is, the more the richer your sample becomes in terms of non-normality. So there are certain types that are maybe more likely than other types, and that kind of make in the way this works, it actually increases the chances that you get 36 types for any one vesicle size. And then I added a memory component, which is like basically carrying over the content from the previous day at a certain rate. And so you can change this number. I put it at 0.05, which means it's retaining like maybe 1/20th of what it had from the previous day, and it's carrying over to the next day. And so this is the code for that. There's a loop that kind of goes through, and actually there are two loops here. That one for the sampling from this distribution, and then the other loop being the memory that carries over, and then you could plot them. And I plotted. So this is what I got. So this mixed distribution, I was able to get it to have sort of, it's for this vesicle size where you never get above 36 kinds. But in this case, I gave it this mixed distribution to draw from. It's actually exceeding 36 occasionally. So you can see that this red line is 36 types, and it's exceeding that occasionally in this series. So this is a pretty long series. This is 10 to the 5th days. You can see that every once on maybe, every few days or maybe every month, it's exceeding this limit. And it's hard to break this down, but you can see that there's some that exceed 36. And so that's, yeah.
I got a question about the graph.
Okay.
Obviously, you don't have 10 to the 5th points on the x-axis.
It's 10. Is it 10 to the 5th?
Well, you said 10 to the 5th. The run was 10 to the 5th.
Yeah.
But the number of points in the graph is not 10 to the 5th.
100,000?
Yeah, I don't see 100,000 points there.
Well, it's really compressed. It's hard to tell.
Yeah,
But
okay,
that's-
my question is this. If you have a point that exceeds 36, does it get rounded down? Or will it show up in the
No,
graph?
it shows up on the y-axis. This is the number of kinds here. So you see the number 36 here, this red line.
Oh, yeah, but there aren't 10 to the 5th points across that are graphed there. Maybe a couple of hundred, but not 10 to
Oh
the
no,
5th.
it's just like it's so dense that you can't see all the detail. But there are 100,000 points
okay.
in here. It's
So I'm
just
plotting all 10 to the 5th,
yeah. It's just really hard to visualize it. Like I can't pull it
Okay.
apart. But actually, that brings up an interesting point, because I can take out pieces of it, and I did that for the second graph. So this is the first 100 days, and as you can see here, you can't really see it here at all, but it doesn't exceed 36. It actually maybe gets up to about 34, and then or 33. So you can see that there's variation over time. You can see it's really non-stationary when you add in the memory and you add in this mixed sample.
Yeah.
You know.
You might add one more graph like that, which is from the section that has only 36.
Yeah, yeah.
Okay.
I have to go now.
All right.
So
All right. Well,
bye,
all right.
and thanks for the interesting session.
Yeah, no problem.
Okay.
All right.
Well, that looks great, Bradley.